What if I told you that right now, as you're just sitting there walking around, your body is burning massive amounts of energy just to prevent you from turning into a rigid, frozen statue?
I mean, it's a brilliant paradox, isn't it? We are so conditioned to think of energy as the, the spark that creates movement.
Right. Like you put gas in a car to make it go,
Exactly. You plug in a blender to make the blades spin.
which totally makes intuitive sense. But today, we're taking a deep dive into a stack of anatomy and physiology notes all about muscle contraction, specifically focusing on ATP, the fundamental energy currency of our cells.
Yeah, and our mission today is to really figure out exactly how energy fuels movement.
Right, and reading through these sources for you, I had this massive aha moment that completely shattered my understanding of the human body. Because the most critical job of energy in your muscles isn't to make you move. It is to force your muscles to let go.
Yeah. Yeah. And getting to the bottom of why that happens requires bridging a pretty mind-bending gap.
Oh, absolutely.
In this deep dive, we are going to connect invisible microscopic biochemistry to massive real-world physical feats. I mean, we're looking at the exact same molecular machinery that allows Usain Bolt to sprint 100 meters in under 10 seconds
Wow.
and allows forensic scientists to accurately estimate a time of death.
Okay, let's unpack this, because before we can appreciate how a muscle actually works, we have to talk about how completely, hilariously wrong the scientific community used to be about it.
Oh, we really were stumbling in the dark for decades. If you look at the early 1900s, physiologists like Walter Fletcher and Frederick Allen Hopkins, they were dissecting muscle tissue, right?
Yeah.
And they kept noticing this buildup of lactic acid. So they made this seemingly logical leap, assuming that lactic acid was the literal trigger causing the muscle to contract.
Which is completely backwards, but I mean, you can see how they got there.
Sure, yeah. They saw a correlation and just assumed causation. Then moving into the 1920s and 30s, this researcher named A.V. Hill comes along.
Right, the thermodynamics guy.
Exactly. He's doing pioneering work, measuring the heat and the mechanical work produced by muscles. But he was basically treat the muscle as a black box.
Meaning he knew what went in and what came out, but had like no idea what was happening inside.
Precisely. He modeled the muscle essentially as an elastic engine. Think of a physical metal spring. You stretch it, it recoils.
Gotcha.
They could measure the heat leaving the system. They could measure the physical work being done. But they had absolutely zero molecular mechanism to explain the how.
Which, honestly, it's hard to blame them when they couldn't physically see the molecules. But that didn't change until 1939, thanks to two Soviet biochemists, Vladimir Engelhardt and Milica Lyubimova.
Right.
And this might be my favorite detail in the notes. Scientists already knew about a protein in muscles called myosin, but they thought myosin was just inert structural scaffolding, like rope.
Yeah, just the physical beams holding the building together.
Right. But Engelhardt and Lyubimova discovered that this structural rope is actually an enzyme. It actively breaks down ATP.
Yeah.
It's like looking at the blueprints of a massive skyscraper and suddenly realizing the steel beams keeping it upright are also the engines powering the elevators, the lights, everything. The scaffolding is the engine.
It's crazy.
But honestly, I struggle to visualize that. How can a structural beam act as an engine?
Well, that was the exact question keeping physiologists awake at night. It was a really startling finding. The tissue itself generates the force, but if it isn't acting like a coiled spring or, a stretched rubber band, what is it actually doing?
Right.
That mystery brings us to 1954, which is arguably the most important year in muscle physiology.
Ah, the sliding filament theory.
Yes. Proposed simultaneously by two completely unrelated pairs of researchers who, incredibly, published back-to-back in the same issue of the journal, Nature, Andrew Huxley and Rolf Niederkirk, alongside Hugh Huxley and Gene Hansen.
Just a phenomenal year to be named Huxley in the sciences.
A vintage year for the Huxleys, truly. Prior to their papers, the dominant idea was the folding theory. Everyone just assumed the protein filaments in the muscle physically shrank, bunching up like an accordion.
But these two teams used interference microscopy, right?
Right, a technique that finally let them look closely at the microscopic repeating units of the muscle, called the sarcomere. They observed these dark bands made of myosin and light bands made of another protein called actin.
And when they triggered the muscle to contract, the dark myosin band didn't shrink one bit. It stayed the exact same length.
Exactly. Only the light actin band got shorter. They prove the filaments don't coil or shrink, they slide past one another. It's similar to interleaving your fingers and pushing your hands together.
Wow.
What's fascinating here is that this core insight, sliding, not shortening, is a 70-year-old model that is held up against decades of scrutiny. In biology, paradigms usually get overturned entirely. This one has only been refined.
Right, like the Limb-Taylor cycle in 1971, which finally mapped out the chemical steps. So let's get into the actual choreography of this microscopic tug of war.
Let's do it.
The notes say it all starts with an electrical zap. A motor neuron releases a neurotransmitter called acetylcholine, which depolarizes the muscle fiber's membrane, the sarcolemma. But you're going to have to translate that for me. Are we talking about a literal electric shock on the skin of the muscle?
That's a really great way to picture it, actually. The sarcolemma is just the outer skin of the muscle cell. Depolarizing just means flipping an electrical switch.
Okay.
Normally, the cell maintains a negative charge inside and a positive charge outside. When the acetylcholine hits the receptors, it opens literal gates, allowing charged sodium ions to rush in, flipping that electrical charge in a wave.
But if that electrical wave is just riding along the outside skin of the muscle, how do the microscopic filaments deep inside the cell know it's time to move?
Because of a brilliant piece of biological plumbing called T-tubules. They are essentially microscopic tunnels that dive from the outer surface deep into the core of the cell.
Like little subways.
Yeah. They carry that electrical waves down into the muscle, acting like an alarm system for a massive internal storage tank called the sarcoplasmic reticulum.
And when the alarm goes off, that tank floods the interior of the cell with calcium ions, which is where the mechanical movement actually begins, because normally a resting muscle is essentially locked down, right?
Right.
The notes describe a rod-shaped protein called tropomyosin that physically blocks the actin filament. Myosin desperately wants to grab onto the actin, but tropomyosin is sitting right in the way, like a physical guardrail.
Exactly. The calcium flood is the master key that removes that guardrail. But it doesn't touch the tropomyosin directly. Instead, the calcium binds to a different regulatory protein called troponin, which sits on top of the guardrail.
So I really want to focus on the how here. How does calcium moving into the cell actually pull a guardrail?
Think of it like molecular magnetism. When the calcium ions bind to the troponin, it changes the electrical charge distribution of that protein.
Oh, interesting.
Yeah, that change forces the troponin to physically contort and twist into a new shape. And because it's attached to the tropomyosin guardrail, that twisting motion physically yanks the guardrail out of the way, exposing the binding sites on the actin.
Okay, so the binding sites are finally exposed. How do we get from there to, like, a bicep curl?
Well, this is step 3, the crossbridge cycle. This is where ATP does the heavy lifting, and it happens in 4 stages. Stage 1, the myosin head reaches up and binds to the exposed actin. We call that connection a crossbridge.
Got it.
Keep in mind, this myosin head is already loaded with potential energy from breaking down a previous ATP molecule, so it's currently holding on to a molecule of ADP and a loose phosphate.
So it attaches. What actually triggers it to pull?
Dropping the baggage. Stage 2 is the power stroke. The myosin head releases that loose phosphate molecule. Releasing that phosphate dramatically shifts the energy state of the protein.
Like a spring uncoiling.
Exactly. The tension built into its folded structure snaps, causing the head to physically pivot. It bends, dragging the actin filament roughly 10 nanometers toward the center of the sarcomere. At the end of that pivot, it drops the ADP.
10 nanometers is unbelievably tiny.
It's practically nothing on its own. But you had to imagine trillions of these cross bridges doing this simultaneously. That microscopic snap scales up to the raw force required to lift a barbell.
Okay, so it attaches, and the chemical release causes it to pivot and pull. Now it needs to let go so it can grab the next spot on the actin rope and pull again.
Which brings us to stage 3, the twist we teased at the very start of this deep dive. To detach from the actin filament, the myosin head absolutely requires a brand new molecule of ATP to come along and bind to it.
Wow.
Yeah, without fresh ATP, the myosin head stays permanently locked onto the actin. It cannot let go.
And if it does get that fresh ATP?
Then stage 4 happens. The myosin breaks that new ATP down into ADP and a loose phosphate. Breaking that chemical bond releases energy, which acts exactly like cocking the hammer of a gun.
Ready to fire again.
Right. It snaps the myosin head back into its starting high-energy position, ready to grab the actin and pivot all over again.
Okay, before we talk about what happens when the ATP runs dry, I want to clarify how the brain scales this whole operation. It's easy to picture one nerve wiring into one muscle fiber, just a neat 1-to-1 ratio. But the sources point out that's a massive oversimplification.
Oh yeah. We operate on motor units, A single motor neuron, plus all the individual muscle fibers it branches out and connects to, forms one motor unit, and the rule is all or nothing.
So it either fires or it doesn't.
Exactly. When that one neuron fires, every single fiber in its unit contracts at maximum capacity.
So how do we control our strength? I mean, why don't I just crush my coffee cup when I pick it up?
By altering how many motor units you recruit and the size of those units. For muscles needing extreme precision, like the muscles tracking this conversation with your eyes, one neuron might only wire into 10 muscle fibers. It's incredibly fine-tuned.
Oh, that makes sense.
But look at a massive postural muscle like the gastrocnemius in your calf. One neuron might innervate 2,000 fibers. When that neuron fires, it's not a delicate adjustment. It's a massive, blunt force contraction.
I love that. Now, I want to go back to stage 3 of the crossbridge cycle, the detachment phase, because here's where it gets really interesting. You mentioned that without a flesh ATP molecule, the myosin head cannot let go of the actin. What happens if the body just completely stops producing ATP?
Well, the most permanent reason your body stops producing ATP is, of course, death. And this molecular mechanism beautifully, or perhaps macabreely, explains rigor mortis.
Right, because when a person dies, they don't stay limp. They turn completely rigidly stiff.
Right. After death, cellular respiration halts, ATP production drops to zero. Meanwhile, the internal plumbing of the cell, like that sarcoplasmic reticulum tank, begins to degrade and break down.
And when that tank breaks down, all the stored calcium leaks out into the cell.
Exactly. The calcium hits the troponin, changes its shape, and yanks the tropomyosin guardrail away. The binding sites on the actin are suddenly exposed.
And the myosin just grabs on.
The myosin heads immediately reach out and bind to the actin. But because there is no ATP being generated to come along and release those heads, they lock. Every muscle in the body becomes rigidly fixed in place. Rigor mortis isn't a special death mechanism. It is literally your normal muscle contraction cycle jammed at step 3.
Forensic scientists actually use this process to estimate the time of death, don't they?
They do, because the biochemistry follows a pretty predictable clock. Rigor mortis generally begins 2 to 6 hours post-mortem, as the last of the ATP reserves are depleted and the calcium starts leaking. It peaks around 12 hours when maximum cross bridges are locked, and it resolves over the next 24 to 84 hours.
But wait, if they aren't making ATP, how does it resolve? I mean, how do they eventually relax?
They don't relax. The stiffness resolves because the muscle proteins themselves, the actin and myosin, are physically decomposing. The structural integrity of the fibers just rots away.
Okay, pulling this back to the living. If a lack of ATP causes muscles to lock up, that implies relaxation in a healthy person isn't just passively turning off a muscle.
No, relaxation is a highly energy-intensive active process. When your brain stops sending the electrical signal, you have to get all that calcium out of the cell's interior so the guardrail can slide back into place. To do that, the body uses something called the Circa pump.
Ah, the sarcoendoplasmic reticulum calcium ATPase.
Try saying that 3 times fast. It basically acts like a biological vacuum cleaner.
Oh.
It physically shoves the calcium back into the storage tank. But here's the catch it has to shove that calcium into a tank that is already stuffed to the brim with calcium. It's pumping against a massive concentration gradient.
So it's like trying to aggressively bail water out of a sinking boat, but the water outside the boat is actively pushing back against your bucket. That has to take a massive amount of energy.
It does. The Circa pump burns huge amounts of ATP to force that calcium back into storage. ATP is just as essential for putting the brakes on a contraction as it is for hitting the gas.
Let's follow that logic. If ATP is required to move, required to detach, and required to pump calcium against pressure, the body must store astronomical amounts of it, like just a giant fuel tank in every bicep.
You would think so. But a resting muscle fiber only stores enough free ATP for about 1 to 2 seconds of maximal contraction.
1 to 2 seconds, that's it.
That's the entire reserve.
That is baffling. That's like having a car with only enough gas in the tank to get out of the driveway. Once you hit the street, the car has to magically drill for oil and refine its own gasoline while driving at highway speeds. How did Usain Bolt sprint for 9.58 seconds if he only had 1 second of fuel?
Because the human body is the most efficient refinery on Earth. It uses 3 distinct energy systems deployed in a specific sequence to bridge that gap.
Okay, walk me through it.
When Bolt explodes off the blocks, he burns that stored ATP instantly. But for the first 8 to 10 seconds of that all-out effort, his muscles seamlessly rely on the phosphocreatine system.
Right. The sources call this the Lohmann reaction, discovered back in 1929.
Yeah, you have a molecule called creatine phosphate sitting in the muscle.
Oh.
It doesn't need oxygen, and it doesn't need to run through complex metabolic assembly lines. It just rapidly donates a phosphate group directly to the used ADP, instantly turning it back into fresh ATP.
Wow.
It's incredibly fast, but the capacity is tiny.
Which perfectly explains the multi-billion-dollar creatine monohydrate supplement industry. Athletes are just trying to slightly overfill that very specific 10-second emergency reserve tank.
Exactly.
But if that first system is so efficient and fast, why doesn't the body just keep doing that forever? Why switch?
Because you simply run out of stored creatine phosphate. Once that taps out around the 10-second mark, the second system kicks in anaerobic glycolysis. Your body starts breaking down stored glucose without using oxygen.
Breaking down glucose without oxygen, but that taps out eventually too, right? I mean, you can't run a marathon on anaerobic glycolysis.
You can't, because breaking down glucose without oxygen creates a buildup of metabolic byproducts, specifically hydrogen ions. That makes the cellular environment too acidic for the enzymes to keep functioning.
So it's short term.
Right. It sustains high-intensity effort for maybe a few minutes. For anything lasting longer than that, your body shifts to the third system, oxidative phosphorylation.
And that takes place inside the mitochondria.
Yes, the mitochondrial pathways. This process requires a steady supply of oxygen, and it is much slower than the first two systems. But the yield is massive.
Huge payout.
Exactly. It produces vastly more ATP per glucose molecule and can also burn fatty acids. This is the slow and steady system that keeps a marathon runner going for hours.
Now, the notes highlight a fascinating medical condition that proves just how distinct these supply chains are. McArdle disease, first described in 1951.
Also known as glycogen storage disease type V. Patients with McArdle disease have a genetic mutation where they lack an enzyme called myophosphorylase.
And what does that do?
Well, without that enzyme, they physically cannot break down their stored glycogen into glucose to feed that second system, the anaerobic glycolysis.
So what happens when they try to, run up a flight of stairs?
They experience almost immediate severe cramping and debilitating fatigue. The entire middle tier of their ATP supply chain is just missing.
Oh, man.
But what's incredible is that many of these patients report getting a literal second wind if they force themselves to wait a few minutes.
Because the third system finally kicks in.
Exactly. The body recognizes the metabolic crisis, bypasses the broken glycolysis system, and shifts directly to burning fatty acids and circulating blood glucose using oxygen in the mitochondria. The cramping stops, and they can continue. It is absolute proof that muscle relies on switchable, distinct ATP supply chains.
That is wild. Now, if anaerobic glycolysis is what taps out after a few minutes of intense exercise, is that where lactic acid comes in? We have to talk about biology's most misunderstood molecule.
Oh, let's clear its name once and for all.
I think almost everyone grew up hearing that lactic acid is the enemy. It's what causes the burning sensation when you lift weights, and it's what causes that terrible stiffness 2 days after a hard workout. But the sources are adamant that is entirely false.
It's one of the most persistent myths in fitness, tracing back to those early 1900s errors. In the 1920s, Otto Meyerhoff did research that eventually won him a Nobel Prize. He proved that lactic acid is just a byproduct of breaking down glycogen under low oxygen conditions.
So it's not bad for you.
Not at all. It's not a toxin, and it doesn't cause cramps. In fact, your body quickly converts it into lactate, shuttles it into the blood, and uses it as a highly efficient fuel source for the heart and liver.
But what about the soreness? Delayed onset muscle soreness, or DOMS. If it's not lactic acid pooling in my legs, what is it?
Lactate clears from your bloodstream within roughly an hour after you stop exercising. The soreness you feel 24 to 72 hours later has zero to do with it.
Nothing at all.
Nothing. DOMS is caused by microscopic damage, literal microtears, to the structural scaffolding of the muscle fibers, particularly when you resist weight on the way down. The pain you feel is the resulting inflammatory response as your body floods the area with nutrients to repair the fibers and make them thicker.
So what does this all mean? We've talked about calcium pumps, molecular guardrails, and switchable energy systems. Why does this deeply microscopic choreography matter for someone just going about their daily life?
Because it dictates almost everything you experience physically. It explains why you pant heavily after a short sprint. You're experiencing EPIC excess post-exercise oxygen consumption.
Catching your breath.
Right. You're breathing hard to literally pay back the oxygen debt required to recharge those fast anaerobic systems. It explains why athletes cramp in the heat. It's rarely dehydration alone. It's often an electrolyte imbalance interfering with that delicate calcium signaling at the neuromuscular junction.
The sources even mention a connection to statins, the cholesterol-lowering drugs.
Yeah, statins can sometimes cause significant muscle pain as a side effect. Why? Because they can inadvertently interfere with the mitochondria's ability to run that third energy system, effectively starving the muscle of ATP. And most importantly, understanding this machinery explains sarcopenia.
The age-related loss of muscle mass.
Yes, as we age, it isn't just that muscle fibers wither away. The mitochondria within them actually become less efficient at producing ATP. It's a compounding cellular energy crisis that severely affects mobility in older adults. Every single step you take relies on this cross-bridge cycle running flawlessly tens of thousands of times a second. If we connect this to the bigger picture, it actually forces us to reconsider what tension even means. Skeletal muscle, your biceps, your quads, burns immense amounts of ATP to hold tension. But the smooth muscle lining your blood vessels has a totally different regulatory mechanism called the latch state.
A latch state.
Yeah. It allows the smooth muscle tissue to clamp down and maintain strong tension for hours using practically zero ATP. The cross bridges bind and then just stay there without needing constant chemical energy to maintain the grip.
Which makes sense. Otherwise, our blood vessels would fatigue like our arms do when holding a heavy box, and our blood pressure would just collapse.
Precisely. It challenges the assumption that physical tension always demands high energy consumption.
To bring this all home, a single muscle contraction is this unbelievable journey. It starts with an electrical wave diving into microscopic tunnels. That wave opens the floodgates for calcium. The calcium uses molecular magnetism to twist the troponin, yanking the tropomyosin guardrails away.
Right.
That allows millions of myosin heads to reach out, grab the actin, snap backward to pull, and then crucially burn a fresh molecule of ATP just to let go and reset. And this is happening continuously across the 650 skeletal muscles in your body, fueled by a three-tiered refinery system that is constantly manufacturing ATP on the fly.
It is a breathtakingly elegant system. But there's one final detail from the notes that I think elevates this from mere mechanics to something almost magical. In 1935, a scientist named Wallace Fenn, who actually studied under A.V. Hill, discovered something called the Fenn effect.
What did he observe?
He discovered that a muscle lifting a heavier load actually releases more chemical energy than a muscle lifting a lighter load over the exact same distance.
Wait, how is that possible? How does a microscopic engine know how steep the hill is before you even press the gas pedal?
That is the ultimate question. The current thinking hints that the physical mechanical strain on the myosin head as it struggles against a heavy weight actually alters its chemical affinity. The physical stress tweaks how fast it binds and burns ATP.
That is wild.
I want you to really ponder that. We are talking about a mindless, microscopic machine that biochemically senses the weight of the dumbbell you are lifting and then adjusts its own energy consumption to match the load, millisecond by millisecond.
It's not just an engine, it's an engine that calculates.
Even at the deepest microscopic level, our bodies are constantly sensing, reacting, and adapting to the physical world around us.
That is incredible and a perfect place to wrap up today's deep dive. Thank you so much for joining us on this journey from the microscopic to the macroscopic. Keep digging, keep asking questions, and stay curious. See you next time.